What if you could change yourself? To become healthier, more beautiful, or even have a different personality? With gene editing technology, we could have the power to do all that. Sounds amazing? I'm not sure we're ready for that yet, and I'm going to tell you why. On November 25th, 2018, a Chinese scientist, Dr. He Jiankui, makes an announcement that shocks the world.
Two beautiful little Chinese girls named Lulu and Nana came crying into the world as healthy as any other babies.
Lulu and Nana's father was HIV positive. Because of the risk of transmitting this to his children during natural conception, Dr. He genetically edited Lulu and Nana's embryos to give them HIV immunity before implanting them into the womb. He announced his medical breakthrough to the world a few weeks later, and this was how the world responded.
I don't think this should have been done.
There's this international outcry, both in China and abroad.
The scientist, He Jiankui, has been sentenced to 3 years in prison. He Jiankui was found guilty of illegal...
This happened around the time I had my first child. I'd want my kids to be immunized against all diseases. So I didn't understand why Dr. He received such a firestorm of criticism. Lulu and Nana are the world's first genetically edited babies. This may have happened in 2018, but the ability to edit genes using CRISPR-Cas9, or CRISPR for short, was actually discovered in 2012. To understand CRISPR's significance, we first need to look at the tiniest building blocks of my body. My DNA is made out of the chemical bases A, C, G, and T. 3 billion of these letters in my body are arranged into a set of instructions to keep me alive. A paragraph of this DNA makes up a gene, and my roughly 20,000 genes are packaged into 46 chromosomes. These 46 chromosomes make up my body's genome. And scientists have figured out how to read all these letters, which tell a unique story about my health, appearance, and personality. I wonder if it can tell me anything I'm unaware of, certain personality traits or diseases I may be predisposed to. Which is why I got my hands on this DNA test kit from Circle DNA. Anyone can buy and do it at home to find out all sorts of information about themselves.
Rub cheeks. Eh? Oh, with the swab. Okay.
It's mind-boggling just what our genes can say about us.
Beyond the medical stuff like my risk for developing cancer, this Circle DNA test claims to be able to tell me things about my personality, physical attributes, and talents.
Sure not.
Will what they find square with what I already know about myself? Or how my environment has shaped me? Guess I'll find out in 18 days. Meanwhile, I'm going to meet someone who not only knows how to read my genome, but also how to change it. Using a tiny pair of molecular scissors called CRISPR-Cas9, the tool that can enter the document of our genomes to cut, copy, and edit our DNA. It could change anything and everything about us. For example, writing away our current and future diseases that have no cure today. Wei Leong studied at the Harvard University lab that first developed this tool.
And it allows us permanent cures to many diseases of genetic and epigenetic origins, and tends to not have cures as of today. So, for example, diseases of the eye, you could deliver this CRISPR-Cas9 into the cells of the eye, and hence, from there, CRISPR-Cas9 would fix the genome, the DNA, hence permanently fixing the disease problems that's associated with that eye disease. But if it's a disease that, say, affects different organs, this could be cancers, or it could be genetic diseases that were inherited. Another approach would be to inject it into the blood, and then it goes everywhere in the body, and then fix the DNA in multiple cells and multiple organs. It could be through aerosols, it could be through nanoparticles. So these are human cells in a plate.
Human cells?
Wei Leong let me get up close and personal with this magical tool.
And then remove it.
So all the magic's happening in there right now.
In the cells, yes.
Right.
Within a few days, CRISPR will remove the diseased gene from the cells I just injected. So if CRISPR has all this potential to cure diseases, I wonder how it can help our fight against our latest pandemic. I didn't know this, but viruses also have genes.
This is COVID-19's genomic sequence.
Knowing this enabled scientists to trace its spread, create diagnostic tests, and work towards a treatment. There are many advantages to CRISPR-based diagnostic tests that can complement existing ones.
For the past 2 days, you've been dealing with a sore throat.
Janice Chen and her team's CRISPR detector test isn't on the market yet, but they're hard at work trying to get it ready.
You may have heard about CRISPR and how it's used in gene editing, but recently we found that the same system can be applied for not just editing DNA, but also detecting any sequence of interest. So in the case of the coronavirus outbreak, we're able to type in letters that will match the coronavirus genome, and that way we can use CRISPR itself to tell us whether or not that virus is present.
So how do I use this?
The user is able to simply take a swab or saliva sample and directly input that into the device, and within 20 minutes you would get a result telling you whether or not you have the coronavirus.
Oh, wow.
In the sense that it's faster, it's cheaper, and it's simpler than the existing method. So right now you would go to a trained healthcare professional to take a sample. That sample would be sent to a centralized laboratory where it's processed, and you would get your result probably 12 to 24 hours after the fact. With CRISPR, you kind of combine the simplicity of, let's say, like a home pregnancy test with the accuracy of a complex molecular lab.
Whoa! If I ever needed to get tested for COVID-19, this is the test I'd want to use. As scientists race to feed the demand for more and better tests, 18 days have finally passed since I sent my saliva to geneticists in Hong Kong. I'm about to find out what they know about me that even I don't.
Hi, guys.
Genetic counselors Arun and Mark are like detectives. They study the clues found in my thousands of genes to piece together a story of me and what I may be passing down to my children.
I would like to straight away drive into the first category, which is basically the family planning. You have a specific genetic mutation in one of the genes, which is HbA1, which is associated with the specific condition, which is called as alpha thalassemia.
So while I don't suffer from alpha thalassemia, I'm a genetic carrier of this incurable blood disorder that could cause bone deformities, delayed growth, and other serious complications in my children. Yikes.
that you have a family history of dementia. And
Yes.
at this point of time, we haven't found any mutations which could drive or increase your risk, so which is a good thing.
Okay.
Actually, the genetic report also look into your physical traits as well. So you're less likely to be thin.
But I mean, all my whole life I've been told by people like what? Hey, have you lost weight?
It's something that you're likely to have in the future, but maybe not now in the current status. Yeah.
If let's say you start having binge eating on different kind of junk food and all of that, definitely you will be the first person to actually end up towards that. Like, of course, you probably had a much healthier lifestyle, and that's probably the reasons why you were able to limit yourself in terms of the overall BMI.
Okay, I'm on the page where it's personality traits now.
So we found that you're someone who tends to be either saying yes to certain things or you're quite diplomatic at times. So one of the things is we found that you have a warrior tolerance. You'd perform much better under stress compared to a lot of other people.
So typically, who would purchase a DNA test like this? And why?
For the couples who are looking forward to start a family, people who are looking to have a personalized dietary and fitness recommendations. Based on your genetics, you can understand what kind of drugs are more efficient for you and what kind of dosage will be more effective for you.
Okay, I was surprised by some of the non-clinical results. Like, for example, my extroversion was extremely extroverted, which I don't find I am. There was only one genetic mutation, and I'm a carrier of alpha thalassemia. So it's good to know.
So, could I use CRISPR to tweak myself? Repair this alpha gene so that my children don't get thalassemia? And maybe extend my lifespan? Scientists have already started looking into the genetic processes of aging and using CRISPR to reverse it. The possibilities are endless. The thing is, I can't do any of that yet. Changing psychological and physical traits like height, stamina, and intelligence through gene editing may be achievable one day. But scientists generally agree that the genetic processes are still too complex to tamper with. As for treating diseases with CRISPR, it's technically possible, but legally not. At least in Singapore. If one were living here or here, one might be able to do CRISPR clinical trials for certain diseases. But many other countries have not officially allowed it. And in consensus with most of the world, Singapore also strictly prohibits the gene editing of human embryos.
Two beautiful little Chinese girls.
Earlier, I learned about the Chinese scientist who genetically edited embryos to be HIV resistant. His breakthrough caused an uproar. But why?
All right, Owen.
Perhaps a bioethicist can enlighten me.
One of the possibilities is that you're intending to edit this gene, CCR5, that would potentially induce immunity to HIV transmission, but you might not edit all of the genes in the embryo. You might only edit some, and that's called mosaicism. And so you might think you're getting immunity, but maybe you won't know. On the other side, you might edit the wrong gene in addition to or instead of the CCR5 gene. And editing the wrong
While CRISPR is much more precise than what we had before, it still could change a piece of DNA it wasn't supposed to. Which is why most countries don't allow tampering with genes at all. Imagine doing a Ctrl-F in your genome to find the DNA you want to edit. This search could generate multiple hits in your genome with similar addresses, when you only want to edit one of them. Accidentally changing the wrong DNA could result in a whole host of dire consequences, like cancer. Although it's more serious when it comes to germline gene editing, which is the editing of an embryo's genes. It means that all genetic changes would be passed down to future generations. So mistakes made would get passed down too. While still a tricky task, any genetic changes made to adults like me would at least end with me and not get passed down to my children. So, to safely treat diseases, gene editing tools would need to be able to find the exact locations to be edited.
Dr. Tan?
I'm meeting another genetic scientist, Dr. Meng Hao, who's trying to do just that.
So our tool is kind of a modification of the original. You can think of it as a chimpanzee.
Chimpanzee?
Chimpanzee walking around with a big pair of scissors.
Oh dear.
This chimpanzee is CRISPR-Cas9. It will be led on the leash to the correct DNA to cut, but could cut the wrong DNA if the leash is too long. What Meng Hao's iCas tool does is to close all the doors between the chimpanzee and the DNA and to install a traffic light. At a red light, the door remains closed. When Meng Hao triggers the green light, the correct door will briefly open, letting the chimpanzee in to cut the correct DNA.
So would you say that this new iCas system that you've developed would make it safer and more accurate for gene editing?
It's really context dependent. For some of the genes, the 100% very clean edits. What people are looking at right now are monogenic diseases, which means that the disease is caused by a single gene. For example, sickle cell anemia, people know which position in the hemoglobin it hits, and if you fix that DNA sequence, for sure, you will get a therapeutic effect. For example, like cancer, it's very hard to use this kind of CRISPR technologies because cancer is a very complex disease with many genetic mutations. Another issue to think about.
So, diseases caused by a single gene like eczema, sickle cell anemia, and muscular dystrophy could be things of the past once this more accurate than CRISPR tool is ready. Meng Hao hopes that will be within the next 5 years. Well, that's not anytime soon. So if we can't legally and safely make changes to our own DNA just yet, are there other things we can edit here in Singapore? I've learned that changing our DNA can cure diseases and one day make enhancements to ourselves and our future generations. But it's still heavily restricted in most of the world, including Singapore. What if, though, humans weren't the only things that we could experiment on?
It's a little worm. It's an orange worm. Is it a bug?
No, it's the mosquito larvae.
Mosquito larvae?
Yeah.
Why is it glowing orange?
We use the CRISPR-Cas9 technology to insert a fluorescent genes into the mosquito genomes. So this will give us the chance to identify the Cas9 mosquito from the non-Cas9 mosquitoes.
The end goal is not to just make mosquitoes glow, right?
No, we're going to use the CRISPR-Cas9 technology to fight the dengue, which is the problem for Singapore here.
Dr. Tyree differentiates his edited mosquitoes from non-edited mosquitoes by making them glow. Singapore's dengue cases have hit an all-time high. And the species of mosquito that spreads it is the Aedes mosquito. Only female Aedes mosquitoes can spread dengue. To stop its spread, Dr. Tyree's team edits the mosquitoes with CRISPR-Cas9, creating the Cas9 mosquito. When Cas9 mosquitoes mate, they only produce male offspring. So not only do they not spread dengue, they inherit the altered genes. And when they mate in the wild, they also only produce males. Eventually, the species will perish.
Now, Professor, you tell me, if this works, we can eliminate dengue. But what are the risks involved?
The one is the mosquito. The Cas9 mosquito might develop resistance. And the second thing is the gene might potentially jump to the other species, like the animal, like the dragonfly, which eat them. The one concern is the gene fragment might integrate in the dragonfly's genomes. There are lots of things need to be done before we can release it there. And we also need to have the approval from the authorities.
Is there a scenario where the mosquitoes might just be wiped out? And does that have implications for the ecosystem?
Yeah, of course, you might cause some change of this ecosystem, but the natural ecosystem, they can adjust to the certain balance again. There's still the hot debate ongoing, whether we should eliminate species. But keep in mind that we only target these Egyptian mosquitoes. There are over 3,500 mosquito species there. We only target a small fraction of mosquito populations, which can transmit the disease to humans.
But mosquitoes and disease aren't the only things that scientists here are experimenting with.
I try to eat as much vegetables as I can. But did you also know that these greens also have genes, which means they can be genetically altered, make them bigger, leafier, and even help you reduce stress?
Like all living organisms, this lettuce plant has genes. To tackle our need for more healthy, locally grown food, Dr. Daisuke Urano spends a lot of time here trying to grow super lettuce. That's lettuce that tastes better, lasts longer, and is resistant to natural disasters.
Some CRISPR vegetables can have higher yield with fewer resources and resistance to stresses like the drought and higher nutritional values. We can increase GABA content. That is the stress reducer for us.
So if I eat this vegetable, I can reduce my stress?
You can get more GABA from the vegetables. Yeah, that potentially reduce your stress level. It totally depends on which gene is edited.
But doctor, why do we need to use gene editing to make our vegetables better? I mean, can't we do it with other agricultural means?
CRISPR-Cas9 is much cheaper, faster, and more safe. Breeding takes a long time to select plants with desired traits, but CRISPR-Cas9 can speed up 10 times or even faster. Fantastic part of the CRISPR-Cas9 technology, we have the capability to change specific traits without changing any other characteristics.
Making our greens bigger and fresher. Will we one day be able to enhance ourselves in similar ways with gene editing? Or at least cure diseases? Until it's fully legalized, do we really need to think about it?
Not so long ago, the same was thought of artificial hearts and creating babies outside of the womb. Now, they are an everyday reality. So we do have to think about how we're going to use it once gene editing becomes perfected and available. And that's why it matters.